Thank you, Professor Jennings. We are going to do a two-person presentation. Would you like to come up also, Dr. Kirkpatrick? I think it may be easier if everybody is up, if you're commenting on the same set of slides and presentation. We're going to continue with the notions of ethical issues in innovation and research with a presentation from two members of the Institute for Philosophy and Public Policy at George Mason University. Primary presentation is going to be given by Dr. Jesse Kirkpatrick and with commentary from Dr. Andrew Light. Thank you.
Great. Well, I think I can speak on Andrew's behalf when I thank the committee for the opportunity to discuss this important and timely topic. As the title suggests, we were requested to speak on the question of if gene drives present novel ethical considerations. And I think that it was with foresight and recognition that, if novel, gene drives might provide impetus for certain precautions or responsibilities to act. So when considering our primary question of do gene drives present novel ethical considerations, we'll be considering three subsidiary questions. The first is, what are the types of ethical considerations related to gene drives? Second, why might gene drives present novel ethical problems? And the third is, could gene drives present novel ethical benefits? So in thinking about ethical considerations related to gene drives, we've identified sort of two types or categories. That is, extrinsic and intrinsic. Now, extrinsic considerations we've heard a lot about already today. These are considerations that are concerned with outcomes or consequences. They involve judgments about the potential risks, harms, hazards, and benefits of a technology. Intrinsic considerations Bruce just sort of raised some potential intrinsic considerations, and these involve judgments and beliefs about the relationship between gene drives and one's deeply held conception of the good. So some examples of extrinsic concerns that we've heard in the first panel are those that are related to human health and well-being or the potential ecological harms of gene drives. And because they are so closely related to consequences and outcomes, extrinsic concerns can often be assuaged by addressing matters of fact related to given concerns. For example, such as providing evidence suggesting that gene drives do not present a threat to human health. An example of intrinsic considerations may be concerns that gene drives represent scientists, quote unquote, playing God or tinkering with nature. So related to intrinsic considerations, unlike questions of human health or environmental risks, and unlike extrinsic considerations, intrinsic objections are not verifiable or testable. They're not because they amount to folk metaphysical intuition that may be based in larger religious, spiritual, or philosophical systems. And more thorough philosophical versions send us down the proverbial metaphysical rabbit hole. Now, an example would be the European experience with genetically modified foods or genetically engineered foods. And this example demonstrates a relationship between worries over extrinsic claims, such as human health and environmental risks, and the development of persistent intrinsic claims, like the claim that GM foods are, quote unquote, unnatural. Now, there are some observations about intrinsic objections. We'll get to why they're particularly important in a moment. But they can lead to what is called a deep disagreement, or deep disagreements. And this is a moral controversy characterized by a metaphysical difference. Again, resolution is not possible by settling a question of fact, by mitigating concerns related to consequences, for example. A classic deep disagreement, the one I just mentioned, is that of genetically modified foods. We can think of other examples, such as abortion in the United States. And the metaphysical question here would be, are genetically modified foods natural? Or are gene drives scientists related to scientists playing God or tinkering with nature in a way that is crossing a red line? And the ethical conclusion, depending on how you slice the metaphysical pie, might be that genetically modified foods are either immoral, that is, that they're unnatural, or morally permissible. And again, these are concerns or considerations that aren't going to be mitigated by appeal to facts or consequences. If one has this metaphysical or ethical red line, very little amount of cajoling or discussing the consequences, backing it up with facts, is going to persuade them. Now, before turning to some of the specific intrinsic and extrinsic concerns associated with gene drives, I first want to state briefly what is novel about them, in our view, and why this novelty may present ethical problems and benefits. So there are three factors that often differentiate technologies and scientific developments with respect to degree, but not necessarily in kind. These are ease of use, speed of change, the regulatory environment. And here, ease of use, when thinking about gene drives, can be characterized by their low cost, the technology that makes them available. I'm thinking of CRISPR-Cas9. They're comparatively low cost to other types of techniques. The fact that they're approaching a sort of simplicity of use, and that the technology consequently can be spread quickly and used by many people. The fact that speed of change is that we're describing that gene editing and the techniques that make it possible, such as CRISPR-Cas9, is a precise, powerful, and effective way to drive a gene through a population at a very quick rate. The regulatory environment, of course, depending upon the technology, is going to be variable. But I think that we can describe the gene drive regulatory environment as being diffuse at the moment. And I think evidenced by the fact that we're meeting here today to discuss it. So in sum is that a full up and running gene drive platform would combine and accelerate these three factors, which taken in sum could result in a combined difference in degree that approaches or amounts to a difference in kind. So these factors that combined are what provide the impetus for urgency surrounding extrinsic and intrinsic considerations. So keeping these three factors in mind, let's now turn to a few ethical problems that gene drives may present. I should say at the outset that many of these extrinsic concerns have been echoed or have been articulated by this panel and in the first meeting that the committee held. I'm only going to touch on two of them in the interest of time. And the first is unintended consequences and accidental release. Right? So, for example, a study that was conducted in 2014 at UCSD published this year, in which gene drive was developed in fruit flies but only used a conventional containment barrier in the lab. Scientists working with pathogens have shown the fallibility of such barrier protocols due to human error, which increases the probability of an accidental release. So as a result, scientists responded quickly, published a piece that noted the shortcomings, and suggested corrective measures. Research centers, such as the Wyss Institute, have taken the lead in developing robust measures to safeguard against accidental release. Heard about what one proposed safeguard could be, and this is reversal drives. In the event of an accidental or intentional release, there could be drives that reverse the original gene drive. I won't go into the details since we've heard about this earlier this morning, but the upshot is that scientists have been able to overwrite and reverse changes made by previous drives, but as we heard earlier, that these drives, there always remains an alteration to the wild type. Right? So the concern here is that because the reversal requires a gene drive, that the CRISPR locus remains. And this causes concern surrounding potential off-target effects, and such as mutagenesis that could be heritable and passed on to future generations. We heard Austin say that he, speak to sort of the redundancy of problems and the concern that reversal drives might not be the best fix because you could be trying to solve a problem with a problem, right? Now, some of the ecological effects could be irreversible. And we also heard about suppression and population modification drives as a potential or holding potentially great hope for the step towards eradicating vector-borne diseases, right, such as malaria. Right, this could take the form of blocking or transmitting the disease or creating a crash drive that drives a species or subspecies to extinction. Now, for obvious reasons, say elimination of a vital food source or the proliferation of invasive pests, this triggers extrinsic concerns, as these ecological impacts could be irreversible, resulting in significant harm. Now, this is an important extrinsic consideration, especially if we consider the possibility that biohacker and community synth biolabs will use this technology. Again, these concerns are not limited to gene drives, and therefore not entirely novel. But when we consider gene drives and technology like CRISPR's ease of use, the speed at which some genes can be driven through wild populations, and the diffuse regulatory environment, these community labs could be ground zero for an accidental release. So a Wilson Center report noted in 2013 that the risks of accidental release related to synth bio and community labs, that the risk was relatively low for these labs to become an incubator for bioterrorists or for accidental release. The report also noted, I think with great foresight, that with changes in technology, so too could the risk level change. So we need regular assessments. The second extrinsic concern I just want to hit on, and has been discussed just a few minutes ago by Dr. Macrino, is the dual-use concerns. So and two here in particular are weaponization of lethal pathogens and biosiege of agricultural crops. But as Daniel Wattendorf of DARPA noted in his address at the first public meeting of this committee in July, gene drive technology raises serious security concerns. We're aware that there are feasibility challenges to these two examples, and it's true that crops will be fairly protected from intentional deleterious gene drives in the developed world. Right? Seed stores are tightly controlled, major crops are grown largely from engineered seeds, but this is not the case in the developing world where controls are weaker. So it's likely that a dedicated, persistent group of individuals or an individual can overcome these challenges. So potential prevention and mitigation strategies have been discussed in the gene drive research community. I applaud them for taking these steps. But we must also keep in mind that many of the encouraging examples we see coming out of the scientific community concerning regulation would perhaps be voluntary, and that regulated gene drives only apply to those who want to follow regulations. The point here is that they're only effective to the extent that they're followed, and that dual use needs to be front center in the debate on gene drives. So turning now to intrinsic objections, sort of the classic case of deep disagreements. How are deep disagreements typically resolved, and how might this apply to gene drives? Well, there's generally two ways. The first is that we stipulate that one view trumps another, right, and accept the consequences of the moral controversy. Example here is the case of abortion in the United States, that an appeal to a woman's right to bodily integrity and self-determination trumps the possible personhood of a fetus. What we have here is one ethical claim or one metaphysical claim, political or social claim, trumping another. And the second is that we allow dissenters to opt out without negative consequences to others. This is the case with genetically modified foods and labeling, segregation in food processing. Another example is kosher cafeterias at universities, right, sort of respecting people's dietary restrictions and allowing them to opt out. The issue that we see is that it is unclear how gene drives will permit opt-outs. Right? In the wild, when gene drives are used, even with full regulatory and legal compliance, we're going to have affected populations that have holdouts, perhaps most acutely at the stage of field trials. And these need to be considered for two important reasons. The one is that we want to value these individuals' reasonable choices and their intrinsic considerations. And the second is that we want to avoid a culture war. So before turning to Andrew and handing the podium over to him, I want to drive home the point that if we don't take extrinsic considerations seriously, the risks, consequences, and potential impacts that could be deleterious resulting from gene drives, such as those in the environment, health, and human health and well-being, then we invite these intrinsic objections, the deep disagreements that might in the face be irresolvable. Andrew?
So I'm not commenting on our presentation. I'm just giving the last part of our presentation. Although I guess I could. I thought it was quite good, didn't you? Right. Our conclusion right now, right, is that in the assignment we were given by the committee is, do gene drives present novel ethical considerations? On the negative side, on the problem side, we'd say, well, technically, no. We don't think there's nothing absolutely novel about gene drive techniques or technology or even the potential of them. But we think taken as a totality, and you look at these three criteria that we're looking at to take as a totality: ease of use, speed of change, and diffuse regulatory environment. These could, taken all together, given how far out gene drives could be with respect to those three criteria, they definitely, we think that there could be definitely public perception, that there's something new and novel that they need to be afraid of. And if we don't take care, and I think we all agree here, we do need to take care of the extrinsic concerns, the risk-based concerns, the concerns for which we can point to protocols and publication standards and all the things that we've talked about all morning long, that we could definitely answer those kinds of things to create a more open environment for innovation. The reason you want to do that, again, is intrinsic worries, because if you don't handle the extrinsic concerns, the intrinsic worries, this just isn't natural to me, I don't want to live in an environment where some new modified mosquito can land on me, can change things about me, could lead to a kind of a pushback against the technology which could definitely hinder innovation. Now, the other reason for doing this, so one might look at sort of the kinds of issues we've raised and been raised in the literature to suggest, for example, that we should slow down research, we should have a moratorium on certain kinds of the next steps along the way in research. We would disagree, and the reason we would disagree is because applying the same kind of criteria, and this is why we're very happy that the committee asked us to look at the ethical considerations and not just the ethical problems, is because there is such potential here, such incredible potential here. And I'm actually going to come back to this one, do the first one, which is just the one that's most discussed in the literature, the potential to create an enormous global public good here, especially with respect to gene drives and the ability to take on vector-borne diseases. Now, I don't have time to go into all the considerations here, but given the potential leapfrog we could get in some of the kinds of biotechnologies that could be produced to deal with these kinds of problems and the kind of harm that you could avoid in the world because of that, and especially, and I'll get back to this in a second, because the abiotic conditions of the world, we know through climate change, are accelerating, is going to accelerate the spread of some of these diseases. Then we're going to need new tools there. And just the need to be, our ability to be able to provide those tools when needed is absolutely essential, I think, and would indicate that we should not slow down research, but protect against the external risks, and also be able to answer the intrinsic questions as best as we possibly could, which will be trickier, and we can come back to that in discussion. With respect to mostly, I've actually spent the last 3 years across the street in the State Department as a climate change negotiator, so this is mostly what's in my mind right now. If we look at the potential harms that can occur in wild populations because of climate change, we're facing an enormous challenge here. And there's a huge range of studies that have been produced on the potential loss of species biodiversity because of climate change. Just this last May, Mark Urban published a very interesting meta-analysis in Science looking at 131 studies, which I think is probably our best guess right now about where we're going in a world where we are at a 4 degrees Celsius rise over pre-industrial levels by the end of the century, we lose something on the order of 1 in 6 species. If there is a value, if you think that there is a value, the kind of value to nature that Bruce was referring to earlier, that there's some kind of value in these species out there for aesthetic reasons, for moral reasons, for spiritual reasons, pick your favorite reason for that, we have to respond to it. And right now, the tools that we have are incredibly clunky, incredibly slow, and don't work very well, mostly involving assisted migration. And a good friend of mine, Ron Sandler at Boston College, wrote a great book on the ethical issues involved in assisted migration, mostly focused on the limitations of what we can do now. And if you look at the many studies that have been done on this, and when I was teaching at the University of Washington, Seattle, I had a lot of great colleagues. Some were looking at reptiles, some were looking at insects, some were looking at amphibians, they were looking at all these different things, especially when we look at the equatorial regions, where we stand to lose the most species, right? Because they've evolved under conditions with a relatively low tolerance towards changes in temperature. Then we're looking at potentially small changes could make it more likely that they could survive in the environment that they're coming into. Strengthening egg shells, strengthening ability for mobility. There's some of the things that was being referred to earlier in ways in which we already know in the literature that you can actually have accidental positive spin-offs in wild populations by creating new tolerances for species that are otherwise at risk. So without using everything at our disposal, I think the choice is global environmental triage at this point. Now, I hope that we get a good climate agreement by the end of this year. That's going to be the most important thing that we can do to try to deal with this problem and get emissions reduced. But given the amount of greenhouse gases that are already in the atmosphere, given the projections in temperature that we're going to see anyway, no matter how aggressive countries move, we're going to need an answer there. We're going to need every tool that we can use to try to mitigate this. And again, the potential with gene drives with respect to these criteria of ease of use, speed of change, and in this case, the uncertain regulatory environment, I think, helps, because it means there's potential for a lot of innovation out there if we create a responsible community of scientists and non-scientists who are watching over where we're going with this technology. We potentially have a moment here where we could actually turn these technologies to focus on priorities like that, where there may be a unique advantage. There may be a unique advantage from this particular set of techniques. We'll only know if we do some intensive research on why these techniques will be needed over the rest of the other alternatives that are out there, and whether or not we can mitigate the risks. Thank you.